Hi everyone, today I want to share a SnakeMake pipeline to do gene ontology analysis. In papers with lots of omics data, it is easy to lose track of how each ontology was generated. This pipeline allows you to document and automate all of your gene ontology analysis. This pipeline only requires a file with a list of transcripts, their fold change and adjusted p-values, and with one click it will give you a publication-ready PDF with the enriched ontologies for the differentially expressed genes. In the first part of the video, I will start by showing you the three components of the pipeline. There is a lot to unpack, so let's move to the first step, that is to get the mapping between gene symbols and our differentially expressed ensemble transcript. To this end, we will use the RMarkdown package to download the most current annotations. So first we specify the dataset that we want to retrieve. In our case, it will be the mouse gene annotation. Then we specify the field that we want to match to our transcript ID. We will be using the MGI symbols that our topGO ontology implementation will take as a parameters. So once we have that, we retrieve the mouse annotation with the use ensemble function over here. And finally, we get the mapping between the gene symbols and transcript using this get biomarked function over here. And then we save this table for later use. The next thing that we want to do is to generate a name vector with the differentially expressed genes to pass to the topGO gene ontology package. In this script, we use the table of transcript with fold chain expression and adjusted p-values as inputs, so are these transcripts over here. And we also request the desired minimum p-value and log fold change to define the differentially expressed genes, and we do that from the beginning over here. The rest of the script converts this table into a name vector. I will not go into the details of the script, but if you want to learn more about it, please check the dedicated video for that with link now in the description. Essentially, the name vector has a name for each position and an assigned number, and this is how it looks like. The transcripts that are not enriched receive a 0, and those that are enriched, like this one over here, gets a 1. With that, we can move to the third part of the pipeline where we use this name vector to run the ontology. In this script, we will generate an ontology notation using the topGO package over here, retrieving the data from the mouse database using this package. At the beginning, we also specify for which adjusted p-value and log fold change we will be using for the input table. We also specify that we want to use the biological processes, BP for short. And for our analysis, we will be also using a classic algorithm and the Fisher test for the statistic. So this script will take us from this input name vector. So then it will generate the ontologies, and then we will run the test, Fisher in our case, and we'll generate a nice table for the result of this test, and we will save it as a nice PDF. So those are the three steps that we are going to put together in our SnakeMake pipeline. But before we jump into the SnakeMake file, I want to show you the configuration file. This configuration file is written in YAML, which allows you to save structured data. So here we define each parameters like the dataset symbols. Most of them are straightforward with the name of the variable and the value that we are assigning to that variable. In the case of the adjusted p-values and log fold change, we have a list of adjusted p-values and log fold change, and this will allow the pipeline to in parallel run the analysis for each of these combinations, and I will show you in a minute how that works. Now let's dive into the snake file itself. So in rule all, we specify what we want, and is these PDFs with the ontologies, and we ask to do it for each combination of log fold change and adjusted p-value. So for that to make more sense, let's look at a graph of the pipeline. We will do that by running this command that will generate the DIG graph. That in our case looks like this. So we first download the symbol, and then when we generate the table that we are going to put into the ontology, we have these different wildcards for the adjusted p-values and the log fold change. We have 2 adjusted p-values, 2 log-fold chains that generates 4 possible combinations, and then we will feed each of these tables into the ontology rule, and this ontology rule will produce the results required for rule all. So if we go back to the snake file, we have the rule all, and then for each of the different steps, like requesting the G-symbol table, the generation of the table to input into the GO implementation, and then the running of the ontology and plotting of the figure. Each of these scripts have their defined rules. But before we finish, I want to show that here when we pass the parameters to this RMarkdown, parameters are specified here in the header. These parameters in the header will be overrun by whatever we define here. Okay, all that sounds good. Let's now run the pipeline. So the pipeline is running. It would not take long to finish. Now it's generating the table, and now it's generating the ontology and the PDF. So now that is completed, and if we check here in our results, we have those 4 tables that we can now open. So if we look at the tables for downregulated and upregulated with value 0.01 as adjusted p-value, we have here these values for the upregulated, cytoplasmic translation on top, and for the downregulated we have regulation of endopeptidase activity. Well, that's it for today. I hope you enjoyed the video. Subscribe if you find this useful. This will make the content more accessible to you and people like you in the future. And I will see you in the next videos.